Hey, it's Tobias. Some of you may know I've been working on this renewable diesel project recently, trying to find the best way to produce diesel from carbon neutral sources instead of refining petroleum. This is a follow-up video to the article I wrote a few weeks ago that detailed 6 of the processes that you can use to get to this renewable diesel product. In that article, I had analyzed that there were 2 processes actually that didn't require a source of green hydrogen, which seemed like a boon because green hydrogen is quite expensive to produce through electrolysis. And so for the last few weeks, I've been diving deeper into these 2 processes. So direct fermentation of hydrocarbons and catalytic conversion of sugars to diesel. First, I'll go through how all of these work and then some of the insights that I took away. The first process that I was looking into right after having written this article is biologically converting sugars into hydrocarbons, which didn't require any source of hydrogen in the process. The way that this works is you start off with corn or any form of biomass, and then you clean this corn to get rid of any dirt. Then there's a process called steeping, where you basically put these corn kernels into a large tank of warm water filled with acid like sulfur dioxide. That loosens the starch in these corn kernels, which makes it easier to break it apart when you go through milling, and then later on centrifugation, which separates the starch and the gluten, which is the source of protein of that corn seed. You can see that we get rid of this pure protein and then take this starch and dry it using heat. Once the starch is dried through heating, you send it to get converted into smaller sugars through enzymes. So starch is basically a long chain of glucose, and what the enzymes will do, they're called amylase enzymes. These enzymes basically cut the bonds between this long glucose chain to give you just the glucose molecule. Unlike the starch chain itself is actually digestible by these bacteria, which you're going to feed your sugars into, and these bacteria are then going to eat it, you could say, and then process it into your desired output, which is the hydrocarbon. So once the amylase enzymes convert the starch into smaller sugars, those sugars then go to fermentation. I was interested in how you could get directly to hydrocarbons from the sugar. And there's actually a company working on this called Global Bioenergies. They're based in France. And I read their patents to understand how this process works. So this is how you can see the process. It all starts in the bioreactor, where you input oxygen and a food source, which is your glucose. And inside of this bioreactor, there's E. coli cells, which are genetically modified. Usually the output of fermentation is something like ethanol, but these bacteria that Global Bioenergies is using, they're genetically modified to give you isobutylene, which is a C4 hydrocarbon. They've modified them to have 2 input-output pathways. So the first one is these E. coli cells can synthesize acetone into 3-hydroxy-3-methyl-butyric acid, and then convert this butyric acid into isobutylene, which is the hydrocarbon, C4 hydrocarbon that you want, and I'll get to that later. In this bioreactor, there's oxygen gas that is inputted, also some acetone to get the isobutylene reaction going, and a food source, which is the glucose. Once they've got butyric acid, they convert that butyric acid into isobutylene. And the reason why acetone is not a required input except for at the beginning, because a byproduct of the isobutylene reaction going from the butyric acid to the isobutylene is to produce acetone. And so this is a self-sustaining acetone reaction. So really all you need is the food source at the beginning, the oxygen, and then a little bit of acetone to get your reaction working. But after that, you don't need to increase the amount of acetone in your reaction. From there, in the bioreactor, your outputs here are isobutylene, acetone, and water vapor, which goes into the absorption column. So at the bottom here, you have oxygen, isobutylene, and acetone that are inputted, and then the oxygen goes up into the atmosphere, but you want to retain your isobutylene and acetone. And so what happens is that water, which is being back propagated from this process, which I'll get to in a minute, this water is coming through, and little droplets of water are basically returning the isobutylene and the acetone into liquid phase. So they come out through the bottom and go into this distillation column. This distillation column is just trying to separate the water and the isobutylene and acetone. The distillation column will heat these, and since acetone and isobutylene evaporate quicker than water, you can get the water that comes out through the bottom and is reused here in the absorption column. So it's a self-sustaining process. The isobutylene and acetone solution go into this second distillation column, which then again heats this solution up, and then the isobutylene evaporates quicker, goes out through the top, which you get your C4 hydrocarbon, and the acetone is returned into the bioreactor to continue that loop of E. coli cells producing butyric acid and then producing the isobutylene. So if you're wondering what isobutylene looks like, this is what it looks like: 1, 2, 3, 4 carbon chain. And so here you're still not done. So what I thought when I was writing that article is that you could directly produce diesel. And I was wrong about that, because this is not diesel. Diesel is a collection of C12 to C16 hydrocarbon chains. This is a C4 hydrocarbon chain. And so you need these larger hydrocarbon chains to be able to have diesel fuel. I mean, there is a way to get to the diesel fuel, which is a process called oligomerization, which is just heat and a catalyst to make these chains longer. But that requires energy, which is what I was trying to avoid with these two processes of not using hydrogen to get to the fuel. One thing that I was wondering is, why can't we get to this C12 or C16 directly? Why does it have to be isobutylene, a C4 carbon chain? The reason why is because what these E. coli cells are basically doing is digesting. This is very high level, but they're basically digesting the glucose, which is a C6 carbon chain, breaking it up, and then pumping out a different hydrocarbon, which is isobutylene. But if you think about it, they're breaking the bonds in the glucose, so you're never going to get a product that is greater than 6 carbons. So my conclusion from this analysis is that the only way to get to diesel from fermentation is through oligomerization of these smaller carbon chains, because most organisms are not able to digest things that are bigger than glucose. So that leads me on to the next one, which is catalytic conversion of sugars. And so this is actually a quite similar process, except it doesn't use biological organisms. It's actually just pure chemistry. So the start of this process is actually quite similar. At the beginning, this processing into the proper feedstock for these enzymes here is exactly the same. Drying of starch, centrifugation to get rid of the protein and separate the starch and the protein is exactly the same. It starts to get different after the enzymes convert starch into sugars. What happens after these amylase enzymes convert your starch into sugar is that you go into a process called aqueous phase reforming. So one thing I learned from working on this is that aqueous phase reforming, or for short APR, is actually a process that researchers use for production of hydrogen. It's been a process that is known for producing hydrogen, not for producing hydrocarbons. I just stumbled upon it because I was looking for how to produce hydrocarbons in any way possible when I was writing that article previously. And this came along, but it's actually a hydrogen production process. What is interesting though is that there is some research about using different sugars instead of alcohols, which is usually what is used in APR, to produce more hydrocarbons instead of hydrogen. So what this means is that usually in APR, most of the research that was done is on using alcohols like sorbitol to produce as much hydrogen as possible, and reactions that would produce alkanes, which is another word for hydrocarbons, were seen as kind of a byproduct that you were trying to avoid. With glucose, because it's less thermodynamically stable in aqueous solution than sorbitol, you actually would get more of these reactions that were, quote, undesirable, but that's actually what I was looking for. So what happens during APR can basically be seen as this process. Glucose plus water gives you hydrogen plus carbon dioxide. All that this is having your sugar in an aqueous solution, so in water, and then reacting it at low temperatures with a catalyst. So here is an example with sorbitol, but it can almost be viewed exactly the same way with glucose. Inside your reactor, which will have water and heat, you actually want two catalysts. So the first is a metal catalyst and the second is an acid catalyst. What your metal catalyst does is that for each carbon-carbon bond that is broken of your molecule, like glucose or sorbitol, there will be one molecule of hydrogen gas, like H2, that will be produced, and one molecule of carbon monoxide or carbon dioxide. On the acid catalyst, your molecule like glucose or sorbitol will experience a dehydration reaction, which is just removing a water molecule. And then this dehydrated molecule that you get, when you add the hydrogen that was produced with the metal catalyst to this, you get alkanes. So what some papers have looked into is that if you balance this correctly, you can actually have the metal catalyst producing the exact right amount of hydrogen for your dehydrated molecule, and then add hydrogen to that and you get alkanes. So you never have to add any hydrogen to the reaction. So for APR here, you can look at the reaction like this, glucose plus water to hydrogen and carbon dioxide. But then there's kind of these byproduct reactions, you could say, but those are the ones that I'm looking for, which is called a water gas shift reaction. So here you have your carbon monoxide plus the hydrogen, that'll give you alkanes and water. So evidently you can just get rid of the water, and then what you're left with here is C1 to 5 hydrocarbons, like I was saying before, because you're starting with glucose, and so you can never have something that has more carbons than the actual molecules which bonds you're breaking. So to get to diesel, you can use oligomerization, which is heat plus catalyst again, and then that'll get you from these, C3, C4 hydrocarbons to the C16s that you want. But also something that I was thinking about here when I was doing this research is that you are not limited, at least to my knowledge, and this is what I'm going to have to look into further. You don't seem to be limited by biological rules like you are in the fermentation, where most microorganisms on Earth can only digest a certain length carbon chain. With this chemical process, APR, I was wondering if you started with a longer chain of glucose and you didn't get your amylase enzyme in the pre-processing to cut it down to C6 chain, what if you cut it down to a C30 chain, and then through these reactions you could break the bonds and hydrogenate your molecule to create your C16, C20, C12 hydrocarbon chain, and then you would no longer require this additional step of oligomerization. So there you have it. That's biological conversion to hydrocarbons and catalytic upgrading of sugars. There's a lot more to figure out, in particular that issue about using longer chain sugars to produce longer chained hydrocarbons. Please feel free to reach out to me on LinkedIn or through email on my website if you have any questions or if there's any resources you have for me. And yeah, next things to look into is if this can work and what the cost factors are. See you soon.